Nucleic acids, namely DNA and RNA, are macromolecules that exist as polymers called polynucleotides. Polynucleotides are composed of monomers known as nucleotides. These are composed of three parts: a nitrogenous base, a 5-carbon sugar, or pentose, and a phosphate group. The portion without a phosphate group is called a nucleoside. Nitrogenous bases differ in chemical groups attached to their rings and can be divided into two groups: pyrimidines and purines. Pyrimidines have 6-membered ring of carbon and nitrogen atoms. Cytosine, thymine, and uracil are all pyrimidines. It is important to note that thymine is only found in DNA, and uracil is only found in RNA. Purines have a 6-membered ring fused to a 5-membered ring. Adenine and guanine are purines. These, along with cytosine, appear in both types of nucleic acid.
Nucleotide sugars are ribose in RNA and deoxyribose in DNA. The key difference between the two is that deoxyribose lacks an oxygen on the second carbon in the ring, denoted as 2', to distinguish it from the second atom of the nitrogenous base, as both are numbered. This completes a nucleotide. To complete the nucleotide, a phosphate group must be attached to the number 5 carbon of the sugar.
Polynucleotides are formed from phosphodiester linkages between adjacent nucleotides. The phosphate group of one nucleotide bonds to the sugar of another, creating a sugar-phosphate backbone for the nucleic acid. The free ends of each polymer are different. One has a phosphate attached to a 5' carbon, and the other has a hydroxyl group on a 3' carbon. These make 5' and 3' ends. Strands have directionality from 5' to 3'.
RNA has only one polynucleotide chain, while DNA has two, which spiral around an imaginary axis, forming a double helix. First proposed by Watson and Crick, the double helix has two backbones running from number 5 to number 3 in opposite directions, forming an antiparallel. On the interior of the strands are nitrogenous bases, or base pairs in DNA. A strand could have a sequence of adenine, guanine, cytosine, adenine, and would be denoted as AGGCA. Only certain bases are compatible with each other in DNA strands. Adenine always pairs with thymine, and guanine always pairs with cytosine. If the previous strand were a DNA strand, the complementary stretch from 3 to 5 on the opposite strand would be TCCGT.
DNA functions as the genetic material that organisms inherit from their parents. Chromosomes contain DNA molecules carrying hundreds of genes, which hold the functions essential for life. DNA is replicated within a cell by messenger RNA, or mRNA, which leads to protein synthesis according to the instructions from DNA strand. This holds limitless implications for the significance of nucleic acids in the functions of life. Thanks for watching, and be sure to find our other videos on macromolecules.